I'm going to talk about genome-wide association. I'm going to talk about the whole human project, but I've also, since I've been working mostly on plants, and since most of the people here are not human geneticists, I will try to also talk a bit about the special challenges when you work on other organisms, and it ties into population genetics in general. Okay, so the basic agenda. Okay, so here's something you want to know, right? How does genetic variation map into phenotypic variation, and how does the environment affect that? And things we might want to do is mapping the causal variants to understand something about the underlying biology. And this is obvious applied interest. And you can also talk about whether you can try to predict the phenotypes based on the genotypes. Okay, so going back really from the beginning. So this is a very old field, of course, and several of the people here, Maria talked about it last week, so I'm going to very quickly zoom through a bit of very textbooky quantitative genetics here. But Mendel discovered genetics in the mid 1800s, and then sort of nothing happened, and then sort of people came back and looked at that around 1900. There was a completely independent school of research by a bunch of mostly British sort of Victorians who were mostly concerned about criminality and where people were stupid in the lower classes and things like this, right? But they started formalizing the obvious observation that like begets like, right? You look more like your parents, you look more like your siblings than you look like something else, right? And you can a very simple way of seeing this is just regressing offspring on parents like this, right? So this is something you can do, and if you have a large class of undergraduates, you just get the height values of their parents and of them, and you see this kind of correlation. So this was observed early on, right? But they didn't know genetics, so they just treated all this statistically. And a lot of, so Francis Galton, Carl Pearson was one of these guys. The Pearson correlation coefficient comes from this kind of work, for instance. Okay. What else about quantitative genetics? You can also start asking questions about, so today, of course, we know that genes underlie this, and here's some other textbooky stuff that I hope everyone is familiar with. If you take two parental populations, now we work on sort of pure lines of something here, right? They differ in mean, you cross something, you get an F1 that's in between. And then if you self-fertilize these, if you work on a plant or if you do brother-sister matings, you tend to get a different kind of distribution. Already from this, you can see that, so this is something that you can get from just simple Mendelian traits, right? And, but already here, you see that something else is going on. It sort of spreads out even more. And you can try from this kind of information to estimate how many genes are involved by this spreading out. And Maria already told you about the breeder's equation, right? That basically, if you take a subsample out here of these guys, right? So select for the tallest one and let that population grow. You get something with a mean that is sort of out there, but another spread and so on. Okay, so the person who sort of then really, so this is really a key paper in genetics, is Fisher's 1918 paper about the correlation between relatives under the superstition of Mendelian inheritance. So what Fisher did, right, is basically, so I think most people know here, but there probably might be some physicists who don't. I mean, the basic idea is he basically took Mendelian genetics, right? So you had this unexplained correlation thing, analysis stuff going on. Then you had Mendelian genetics, where there was a mechanism for transmission, but that was all based on a single locus, right? So what Fisher basically said, okay, look, now assume that there are lots and lots of these genes, right? Jenny just put that into basically a linear model, did a whole bunch of linear algebra, although that wasn't really known as that yet, and showed that you can explain almost all these results. You can explain the correlation between relatives just by supposing that the trait is actually due to a very large number of Mendelian genes that have a very small effect. So so a bunch of very things comes from this. So he basically formalized that you have for each genotype, right? You have, the big A genotype pulls you A amount of whatever measurement it is in that direction, the other direction. And if there's no dominance, the heterozygote would be 0 right in the middle. D here refers to like a slight displacement one way or the other. Frequencies like this, you can calculate the population mean. I think Maria said most of this already. So yeah, they introduced notions like dominance, right, which reflects the deviation from additivity between alleles. So it's all a very, very, very linear framework, right? Epistasis then is a deviation from additivity between loci. So in the simplest model, there's no dominance, right? Allelic effects add up. It's just like zeros and ones. You can just count them and figure out the phenotype. And you can count between loci. You can count among chromosomes as well, right? Okay. Yeah, you can define things like the average effect of an allele, which is the mean deviation that basically, so a notion here is like, what is the effect of a particular allele given the genetic background, if you can just swap it from the other, right? I mean, this is, yeah, okay. And other games, and Maria already said most of this, you can also then take the whole phenotypic variance, you can decompose it into that, which can be attributed to genetics. And there's various interaction effects, right? So you can divide it into the additive part, the dominance part, and so on and so forth. Anyway, I'm not going to go through it very quickly. But one important concept that ties into this is the heritability, which is basically the proportion of the phenotypic variance that can be attributed to genetic factors, either in the broad sense, I mean, all these various things, dominance and so on, or in the narrow sense, the additive bit. And as Maria showed last week, that's the bit that actually responds to selection. And it turns out to be the slope of that. You can estimate it from the slope of this equation here, right? It basically shows you how well can you predict the offspring in a linear fashion from the parents. And if you just do a simple selection, that also translates into how well selection works, right? If it's 100% heritable, then if you take a new population, remember this, if you take something from the population out here, if you take the top 10% and they're like 10 centimeters taller than these guys, right? Then the new population will be 10 centimeters taller as well if there's 100% heritability. Otherwise, the response will be proportionally lower, right? It's just in a linear fashion. Okay. Right, so that was just a bit of quantitative genetics stuff. But now I'm going to talk about how you map genes. How do we map genes? Well, to be honest, it's been a while since I did an undergraduate textbook, but when I was an undergraduate, undergraduates had to do a lot of this, right? This is from human genetics, and you probably still are terrorized by this because textbooks never change, right? Even though people don't really do too much of this anymore. There was a lot of linkage mapping, right? So what you can do, so this is a human pedigree, you have dad and mom, and then you go down here and see who has a phenotype, and that's the black, right? And then you basically try to figure out whether a trait is co-inherited with particular markers, right? You can figure out how a trait is inherited this way and so forth. Okay, another way you can do it is if you work on an experimental organism, you do a genetic cross. In this case, you have a population here of plants with hairy leaves and those without hairy leaves, and you cross them and you get something in between. And then if you take these guys and self them and inbreed them for a number of generations, you get a population that is homozygous effectively again, so that we can ignore it. So I'm just drawing one chromosome, but it's basically segregated. You basically have a mixture of the two parental genomes, and then the name of the game is simply to find correlations, right? Correlations between, in this case, so the less you're supposed to see here that the less hairy guys all share this tip of the chromosome, right? So you'd imagine that there's some factor that affects hairiness that's sitting up there. Now, okay, so there's a huge literature on all this, and I'm blowing over it because, in a sense, that's all it is, right? You're just looking for correlation between genotype and phenotype. And in this case, with this kind of thing, correlation actually means causation, right? Because there is no other possible explanation. Of course, genomes don't come in. Okay, so now, genomes don't come in blue and orange, of course, right? How do you actually do this? Well, you need to have genetic markers, right? And this is also, the textbooks are full of this stuff, but in some sense that's completely trivial, because actually these days genomes do come in blue and orange. The way we do genetics crosses in Arabidopsis, we make a population of this, and then we sequence everyone. So then we have, in any cross like this, several 100,000 SNPs, and you can with certainty attribute any segment of the chromosome to exactly where it came from, right? So that completely, there is a large and somewhat complicated literature on mapping functions and stuff like this, which is basically about, so the way people used to do this is they had markers all over the chromosome, and then you basically have to slide a likelihood model for what if the gene is somewhere between these markers, right? And like, you know, basically model what could there be between these, given that we only see what's going on here. But who cares today? Because you just brute forces, you just sequence everything, you know exactly what's every position, and then it just becomes a complete, you know, correlation exercise. However, let's see, what's on the next slide? Yeah, right. The problem, right? So that has changed completely, and so then, and the other thing. So there are really two things you need. You need some way of modeling what's being transmitted, right? But actually, in a modern setting like this, you don't need to model it because you can see it directly, right? You can just see what, and the odds are pretty good that you often will actually have the causal variant in there, right? So then there's nothing, you know, nothing to model at all. Yes. So if you do an experiment like this, if you just want to map a trait in Arabidopsis of flies, right? This is dead easy, right? You just take two parents that differ, you cross them, you make a very large number of offspring, and phenotype those as well, and then sequence all of them, and then you just figure out which particular genetic variant is correlated with this, right? Right. So the way people used to do that is by just having a small number of markers, and sort of then you would run a likelihood function to see, you know, what if there was a marker between these things, where, you know, you have to, so there are mapping functions, there's a Lander and Botstein and stuff like that. You basically see where could the marker be, you model how recombination must have happened and so on. But increasingly, I think this is, at least for model organisms, there's no point in doing this because you really do sequence everything. Now, so things I'm glossing over here is that you're still in a model for genotype to phenotype, right? Because of course there can be many things segregating at the same time, and that can screw you up, and I'll get back to that later in this talk. The main point here is what happened about 20 years ago is that there was a revolution in technology, which generally is what drives biology forward. And that so it became possible to generate infinitely many markers, right? I mean, if you look at, in this kind of experiment, so even if you can paint these chromosomes completely, the resolution in such a genetic cross, or in a pedigree, like the human pedigree I showed before, is extremely limited because the only information about the position here comes from the number of crossovers that happened here. And basically, you know, half of them happen in this F1 generation, and then as things become homozygous, there's more recombination. But in the end, all you do here is you identify a piece of the chromosome that is quite large. Let's see what's in my next things. Yes, right. So this is what I'm going to say. So resolution was limited by markers, right? Yeah. Okay. So let me go on about this for a bit more. So historically, that the fact that you had such a big thing didn't really matter because there were no genetic markers to get you closer anyway. Today, you have infinitely many markers effectively, right? And in fact, you can even have the causal site in there. But that doesn't help if you have the causal site in there, because presumably the thing that actually causes hairiness is in here. But so will, you know, 10,000 other markers be. So you have no way of telling which one is which. So what can you do? Well, in this kind of design, you can always increase the sample size, right? So the resolution here is linearly dependent on how many things you, how big your population is, right? Because the more of these guys you do, you know, the finer the interval. It's just more recombination, and that's completely linear. And that works for an experimental cross, but of course this gets impractical. Like actually, it's not the genotyping of that. What typically gets impractical is just rearing that many individuals can be extremely difficult. But you know, in principle, you can do this. But of course, in human genetics, this was never an option, because you can't, you know, humans, you can't we don't have inbred lines. If I want to rear 10,000 offspring of an Arabidopsis cross, I just do it, right? And the same thing for flies, but humans do not have 10,000 children. And so a lot of these human genetic studies, I mean, this is the reason so much of that work was done in places like Utah, partly because people actually keep track of, I mean, to map things in human genetics, it's even more complicated, right? Because typically you don't have two parents like this. What you actually had is some kind of disease. You know, people get sick, they go to the doctor. If the doctor has seen a lot of patients and they read other papers and so on, they start recognizing, oh, this is such and such syndrome, right? So Huntington or whatever, right? So Huntington, and it gets named after the doctor who just realized, oh, this is one thing, right? All these people seem to have the same thing. Then you need to find a family in which this thing is segregated, or many families, and you put all that stuff together. And it gets really, really complicated, right? You have people that come to the hospital, you need to identify which ones of those have informative families. Those families are never very large, so you can't map things very, very finely, and so on. But, okay, this... so I'll get to the alternative here in a second. So the approach here, what I just said, also has very high power, right? Because, you know, at least in this mapping thing, because you set up a population where the causal alleles have very high frequency, right? So assuming that you have a large enough sample size, you'd always succeed in mapping things. And if you do experimental crosses, you can control the environment, which of course you cannot do for humans. Right, yes. Okay. Right, so the problem here is the sample size, right? So a real question here is, what if there are, you know, what do we think, how big do you think these alleles are? So if you take Fisher's model, right? Here's a very, very important statement, right? Which should really be sort of printed in gigantic letters, and everyone everyone who does anything should be forced to sort of memorize this. So it's very important to remember that a quantitative trait can be extremely highly heritable, and yet that can have the property that there is no single polymorphism, no single contributing factor that is large enough effect to be mapped or studied, right? I mean, this is very important. I'm going to get to this in the human genetic setting in a while, right? There are many, many traits. Like human height is the classic one, that is highly heritable, right? That doesn't mean that you'll be able to find the genes for it, because if it's spread, if that heritability is due to a very large number of genes, there is a mass action effect here, right? You can in aggregate see that if you're more similar to your brother, right, than you are to your cousin, but that's because you share twice as much of the genome, right? And that is a lot of polymorphisms. Yeah, in fact, so if you're as old as I am, in the early days of QTL mapping, there were many of the classically trained quantitative geneticists who poo-pooed the whole idea, because they were so wedded to this infinitesimal model that they thought the whole idea that you'd be able to map genes for quantitative traits was ludicrous. They were, of course, wrong, because you know, the truth is somewhere in between here. Right. Okay, so genome-wide association studies. Right. So the Human Genome Project. So there's a history for all of this, where this came from. With the sequencing of the human genome, which was like, you know, a monumental undertaking, you sequence one human genome, people then started discovering markers everywhere. So there were SNPs all over the genome, right? So suddenly, all this stuff in the textbook about, you know, microsatellites and mini satellites and whatever people used to use, right? There were all these markers that were used for these pedigree. And suddenly that wasn't limited at all. People discovered SNPs. This was, like I said, and there are many population geneticists who own the livelihood to the fact that human geneticists discovered SNPs and linkage equilibrium and didn't know what to do with it. Then it became obvious to a whole bunch of people, and you know, Eric Lander was one of the people who pushed this very, very strongly. Like, look, in the end, all we're doing is correlating markers with a phenotype in a pedigree, or you know, where you look at co-transmission of things in this pedigree or in a cross or whatever. Why not just do that in the whole population, right? If you want to find an allele for blue eyes, right, just genotype a large population of blue-eyed and brown-eyed people and see what is it that all the blue-eyed people have that the brown-eyed people don't have. It's a pretty obvious idea, right? And why would this work? Well, so it was genome-wide association studies. I mean, so basically you just go across the whole genome and look for an association. Why would this And it's exactly the same idea as in a cross, right? Except the study population is different. And how is it different? Well, when you make a cross, you start out with 2 different parents, so all the allele frequencies are going to be at very high frequency, right? And you have these large linkage blocks, right? So you have big LD, which is the reason you don't get any resolution. But if you remember my lectures about the ancestral recombination graph, in a large population, right, linkage equilibrium correlations between alleles are actually only seen over very small distances. In humans, you have LD decaying over maybe on the order of 20 kb or something like this, right? So actually, if you can actually, you have very small blocks, so there's a chance of actually finding markers that are tightly linked to the causal sites. Now, and of course, you can even find the causal sites itself. But in the early days, and this paper is from 1999, this was unreasonable. So because it was still very, very expensive to genotype markers. So Krugler here, for instance, wrote a paper basically trying to do a power calculation, like, okay, how many SNPs do we need to genotype, given what we know about linkage equilibrium, to have an appreciable chance of finding ones that are correlated with the disease, right? And then there was like 10 years of this huge literature on haplotype blocks and how much, how do you pick tag SNPs to do this, that, and the other. And in the end, so he here proposed 500,000. That was sort of a good number, because that meant that people like Lander and Collins could go to Congress and say, well, actually, we only need to do 500,000. We can afford to do this, right? It will cost so and so many hundred millions, right? Because if someone had said, oh no, we need to sequence the whole human genome, we're like, forget it, that's impossible, we can't do it. Of course, today people actually do sequence essentially the whole human genome anyway, right? I mean, not quite yet, but it's getting there very quickly. And in organisms like Arabidopsis and Drosophila, this is exactly what you would do. You would sequence the whole genome. You don't care about selecting any SNPs because it's trivial, right? So this is something I have learned in my life, at least. Never bet against technology, right? Things are always going to be cheaper. You just imagine the biggest experiment you can imagine and just start doing it, because by the time you get there, it'll be, if it relies on technology, other things like field work do not become any easier. Anyway, today in human GWAS, people use at least 1 million SNPs, not 500,000. But broadly speaking, this was right. So, yes, hello. Whoops. Yeah, okay. So the promise of this was basically, because linkage equilibrium, and everyone understands this now, I hope, linkage equilibrium decays very quickly. So if you find an association in a general population, like in the crossing population, you have these big blocks that came from mom or dad, right? And they're megabases in size, right? Remember recombination? I told you like a centimorgan, which is 1% crossing over, is about a megabase in humans, right? And in a cross or a pedigree, where you're talking with hundreds of thousands of individuals, you're obviously going to be dealing with recombination no more closely than on the centimorgan scales. You'll be dealing with megabase chunks. You will never be able to map anything more closely than that, because in that population, right, you will have linkage equilibrium over very big pieces. However, in a big outbred population, like for example, if you should take the eye color example, right? If it were the case that all blue-eyed people were blue-eyed for the same reason, like some loss of function in some pigment allele, right? That must have happened a very long time ago, because that's a pretty widespread allele. And the haplotype sharing around that allele by now is going to be very small, because there will have been hundreds of thousands of years for this thing to recombine with other alleles. So you will be very close if you find a correlation. Okay, so one of the promise of doing this would be that we have gene level resolution. Okay, and that's great no matter what you do, experimental organisms or humans. But so we were doing exactly the same thing. I was trying to drive the same thing in Arabidopsis at the same time as people were doing this in humans, and people were doing the same thing in maize, and so on and so forth. But of course, humans were way ahead because they had the money to actually do the whole genome and so on. Another big advantage, and this is mostly for human genetics, but it's an extremely big practical advantage. As I said before, if you want to map something in flies or in crop or whatever, you just make a cross, right? You can't do that in humans. You have to do pedigrees, and that's really, really hard. You have to take sick people, they go to the hospital, then you have to identify families in which this thing is segregating so you can map it, blah, blah, blah. This is very, very difficult and doesn't always work. And it clearly can only work for traits that have such a strong inheritance that you can see them segregating in families, right? There are many traits that are, so this is, there are many things with it. There's the online Mendelian inheritance in man with like a couple of thousand diseases that have been mapped using these linkage studies. But these are all diseases that are very severe, very rare, have a very simple inheritance, and you can see them segregating in families. So, think about other traits in humans. Things like height, for instance, we know it's highly heritable, but it doesn't segregate in families, right? It's not like if you have a whole bunch of siblings, so some of them are going to be really small, everyone is sort of smeared out there. And then there are some traits, and I'll get back to that later, things like skin color, where you actually, if you have a big enough, you can sometimes see it segregating in families, right? Because you can have siblings that are actually quite different in skin color, and so on. So they're different. So if you don't have to rely on pedigree, so that was the pedigree idea. Now in the GWAS setting, instead, what you do, right? We're just going to find people, like I said, blue-eyed, brown-eyed people. You just take people that are sick, take people that are healthy, sequence both of them, and compare. This is beautiful, right? Because here you don't need to go and dig up any pedigrees and try to infer things. You can just take all the cancer patients with a certain cancer that come to a hospital, right? Genotype them, identify a control group, and we'll talk about that later, what the control group means, and bang, logistically, this is completely different. Okay, does it work? So this was tested, this was put to the test. The Human HapMap Project, so remember that paper was from 1999, less than 10 years later, people had done the experiment, right? This is probably this is the first really big paper that did this. And so this is essentially the first genome-wide association paper. So they had, to do this, what you first need to do, right, is the Human HapMap Project. They took 96 individuals, or sort of population sample from different, there was the Yorubans, they had Chinese, they had Chinese, Koreans, Japanese, there was a bit of mixture, and then Europeans from Utah. Sequence these guys, develop markers, develop a SNP chip, genotype things, right? And what they do with this then is, there are 14,000 cases of 7 common diseases and 3,000 controls. And to demystify this completely, this is the key figure. Here are diseases: bipolar disorder, coronary artery disease, Crohn's disease, which is an autoimmune stomach horrible thing, hypertension, rheumatoid arthritis, type 1 and 2 diabetes. All right, what are we looking at? So I'm going to show you lots of these things. So these are chromosomes going this way, right? So all the human chromosomes, that's that scale. And if you go along here, each dot is a SNP. So there are 500,000 of these. And on this axis, you have the negative log p-value of association, right? And you can essentially think of this as doing a chi-square test, right? So this is basically 500,000 times 7 chi-square tests, right? It's not necessarily just a chi-square test, but it's not that different. It's just a test of association. One marker, tested association, right? So you have a SNP marker 0, 1, you have disease status, sick or not sick, right? They had 3,000 controls, and then a couple of thousand patients for each of these diseases, right? So you have a contingency table, you calculate the p-value, and you plot the p-value like this, right? And you see a distribution of p-values, and what you do, and that, under the null hypothesis that most of the genome isn't involved, that should just be, a completely uniform, right? But you obviously have to, here's a case, and there's a big literature in this, what do you do with multiple comparisons, right? Because there's a lot of comparisons here, so how to correct multiple testing here is a problem. Yes? You can do either, but you have to estimate the effect sizes then, right? And these are, yeah, sure, absolutely. Why do you do that? Well, I mean, so these are basically, these first are binary traits, right? Most of these. So these aren't quantitative phenotypes. So there's no, the effect size then would be in terms of the odds, the increased risk factor. Yeah, sometimes people do that as well. I think it's just, yeah, you can do it either way. We sometimes plot effect sizes as well, right? I guess the p-values are sort of have an easier interpretation. The other one is more model dependent in some sense, right? You know, how you estimate the effect sizes. It's harder to compare between traits also, I guess, maybe. Depends on how you normalize it and so on. Yeah. But, you know, then you would sort of, the green ones here are the ones that are deemed to be genome-wide significant. And then you see these spikes of things shooting up here, right? So in some sense, bingo, right? It works in the sense that you have these, these are like, what each of these spikes, if you zoom in on that, that will be a region of like 100 kb region, that will be a sharp spike of association. And that is basically the SNPs that are very closely linked to a causal variant. And the linkage equilibrium drops off very quickly. If you do these things, I don't have a figure of that in here. If you showed one of these QTL mapping or one of these pedigree mapping functions, right? Typically what you have there is log curves of where things are covering like entire chromosomes, right? So you have no idea where the gene is. You know, here the resolution is exquisite, right? And you see there are spikes everywhere here. Well, not everywhere actually. So there are a couple of interesting things that you can notice. First of all, there is a difference, right? Like for instance, bipolar disorder, there's very little. Whereas for type 1 diabetes and for Crohn's disease, there's quite a lot. So this was something that was observed early on. Like for these behavioral diseases, you know, they were not terribly successful at first. Are they somehow different? I mean, so you can imagine, well, let's have a guess. Why do you think that is? There are basically, there are several possible explanations, but let's have a few. Why do you think it might be harder to map bipolar disorder or schizophrenia or depression than say, rheumatoid arthritis or Crohn's disease? Yeah, that's one. So the heritability is lower because it has a bigger environmental effect. Any others? Right, because that would basically, you know, remember what I said here, this is just a correlation exercise, right? So this will only work to the extent that there is a correlation, and that correlation is, you know, obviously lessened by how much environmental noise there is, right? What else could affect this? A little bit louder, please. So you talk about the different alleles, yeah? No? Yes, yes, exactly, right? So the genetic architecture of the trait has a huge effect, right? If a trait, I mean, this is like Fisher's model. I mean, this would not work if this was completely quantitative, right? And you know, there was no significant effects of any kind, right? They have to be big enough, right? And of course, this is important, right? The correlation here, this is going to depend on both, you know, the additive effect, right? We call that alpha. And so the effect size, and also, this is very important, x, 1 minus x, right? So, yeah, so basically, you know, the p-val, actually the negative log p-val, is going to be sort of proportional to this, right? Where this is the allele frequency, right? So, yeah, and this is, you know, effect. You know, because if you have a very big effect allele, so many of these Mendelian diseases that I mentioned that have been mapped in pedigree model wouldn't necessarily, you know, would be very difficult to map in the general population, right? Because they're so incredibly rare. I mean, so most of the things that map that way, you know, you map a disease, you find an allele, but it turns out that it only affects 184 people in Eastern Finland or something like that, right? Because it's a very rare thing. Same thing for, you know, like the breast cancer, the BRCA mutations and so on. I mean, if there are certain alleles that have an incredibly elevated risk, you know, to the extent that people, you know, choose to have preventive surgery if they have a particular genotype, but they don't explain much of breast cancer epidemiology in the population because that's due to lots and lots of different alleles. And that also ties into, so right, so that's the genetic architecture, but there's something else that affects the genetic architecture of a trait. And that's how it's defined, right? You know, some traits are complex. I mean, it's been said that a complex trait is just a poorly defined phenotype. because probably, if you can probably think about this, I mean, you know, Crohn's disease is an autoimmune thing that basically your sort of guts basically ends up attacking itself. And, you know, I don't remember the details of this, but it's a relatively well-defined thing, and you can imagine that there's a pretty straightforward genetic basis for this. But a trait, well, you know, the reason height is a complex trait is because, you know, everything affects height, right? I mean, you know, our immune system will affect height because how well we defend against pathogens will affect how big we grow. Our food uptake will affect height. Everything will affect height. And if you take these behavioral disorders, you know, it's very difficult to figure out. I mean, the diagnosis there is very uncertain, right? I mean, different doctors will have different diagnoses. Such and such a person is schizophrenic or not. So and such a person is depressed. You know, this is extremely difficult to standardize this. So probably what's going on for many of these things is that you actually confound a whole bunch of different underlying molecular traits that leads to a phenotype that somehow by some doctors get called bipolar disorder, right? So then it gets more diffused because of that. So once you have defined the phenotype that way, the effect of any different allele is going to be very small because it only affects a subset of what's going on. And so but what I was going to say, so that was from these first findings. Now we know that there is nothing different here. Indeed, if what's ended up happening is that even for almost all, I think almost all of these behavioral disorders now, progress has been made. But what it took was huge international consortia, like people laying aside their rivalries, combining all the samples into huge, so bringing up the huge sample sizes, and then suddenly there's progress. You know, suddenly you start seeing signals for these things as well, simply because you increase the sample size. It also, the other thing that really helps is refining the study. Like I heard a talk by Jonathan Flint. They published one of the first sort of genome-wide significant things for depression recently. That was done in China, actually. That was years of effort. So the problem with depression is there's so many things that predispose you to depression, you know, alcohol, you know, so there's it's pretty ugly. I mean, you know, there's all kinds of abuse. Sexual abuse as a child is a major risk factor for depression and so on. So this was a study that was done all on middle-aged Chinese women. So they took it all to China. It was 10 years of collecting data, educating doctors to ask the same kind of questionnaire. And you can imagine just how difficult it is to go out and actually get accurate data and like actually get people to truthfully answer whether they were abused as a child and things like this. You know, this takes an enormous effort to get these kind of data. But in the end, they actually had some results. So they picked middle-aged Chinese women because basically they would not have smoked or done much drinking, right? Unlike the men, and you know, sort of there's a cultural thing and so on. Anyway, whatever. Enough about that. Another thing I just wanted to point out here before I moved on is this. You notice here, there's a big peak for both type 1 diabetes and rheumatoid arthritis. Anyone want to venture a guess for why that is? So here's an overlap in genetics. This is just a cute little fact, but what kind of diseases are these? You know, there's type 1 and type 2 diabetes. Type 2 diabetes is if you eat too much. Type 1 is the one you get as a child, basically. Autoimmune diseases. This is the MHC. And this was well known before, that their immune system leads to this thing. These are basically the immune system attacking your own body in some sense, and there are allelic variants for that. Okay. Right, okay, so great. So this was like 2007, but, so I remember being, yeah. Sorry? I'll get to height. For height, you would find absolutely nothing with this kind of study. And the first ones were also done, these are all case control studies, right? Here they're taking diseases and control, right? The first studies of quantitative traits where you actually have a measurement instead, right? Where you wouldn't run a chi-square test, you would run like a t-test for each thing. You know, regression came a bit later, but I'll get to those in a second. Yes? Yeah. False positives, we'll get into false positives too. I mean, so this is a huge issue, but I'll talk about that in a second. I think these are pretty conservatively defined here. No, they were probably real, right? It's, you know, it's, I can't, you know, I don't know whether these particular ones were real. You know, I'm not an expert of bipolar disorder or so on and so forth. But I mean, yes, there can be false positives, and you know, potential sources of that would just be statistical flukes, right? But I will tell you about a much more serious one later on, which becomes very important when you do, certainly outside humans, but anytime you do cohort studies instead of these cases and controls. Yes. Right, okay, so I remember being at the Cold Spring Harbor meetings when they were first sort of discussing these things. And, you know, it was like, wow, this stuff is really cool. But there was like a murmur in the room, right? Because basically, something that quickly became apparent is like, wait a minute, something is wrong here. Because basically they had, you know, these traits, right? And you have the whole genome. So the problem was that you don't explain the variation. So now we're going to get to height here. The paper that really brought this home was this one. So genome-wide association analysis identifies 20 loci that influence adult height. And this was in, well, I should put the date on there. This was also in the 2000s sometime. So here now they had brought the thing out to 16,000 samples. So yeah, so for the, I mean, this is what I said with like breast cancer and stuff like that. You find a lot of associations, but if you try, can you explain all the cases, all the sick people this way? And the answer is not. You can make this more quantifiable with the height data. So here they had 16,000 samples, and with genome-wide significance, you know, p-value less than 10 to the minus 7, or 10 of them reaching 10 to the minus 10 insignificant. These 20 SNPs, it's like, yay, we found 20 SNPs. They explain 3% of height variation. Okay, now that has since been updated. Yeah, so I just on the flight over here, I was just getting, so the latest one on bioRxiv that just was posted from many of the same guys from there's this giant consortium now. They have upped now the sample size to 700,000 individuals. Now they identify 3,290 significant SNPs. That together explain 24.6% of the variation. Okay, so, well, that looks more respectable, but the problem is, of course, this, that height has a heritability of 80%. Right? So, and so this goes to many of the disease things too, right? So basically, if you've seen, perhaps there was a movie called Gattaca, you know, about sort of science fiction or anything, that you let people reproduce based on their genotype, and there's sort of this, you know, you're going to predict who's going to get sick, and people will be denied insurance coverage because they don't, you know, blah, blah, blah. You can, you know, people are going to choose their babies. I mean, this is all bullshit, right? I mean, if you want to have a tall baby, just have sex with somebody tall. I mean, you know, quantitative genetics works. The heritability is extremely high. Doing it from SNPs, right? Even with these huge sample sizes, they are still only explaining, you know, 25% of the variation. We can explain 80% of the variation by just regressing parents and on offspring, right? So what is going on here? So this then led to this whole thing about finding the so-called missing heritability. And so that was the thing, right? We know that many of these traits, even some of these, you know, behavioral traits, as schizophrenia has a very high heritability. You look now at the whole genetic variation, why can't we find the variance? Right? So blah, 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 lots of, you know, news and so on, right? So the simplest explanation for this, and this is another very important paper, I think, from Peter Vicious' group, that basically actually show that, in fact, common SNPs do explain a large proportion of the variability. So they reanalyzed some of these height data in a different way. And here they show that 45% of the variance can in fact be explained by considering all the SNPs simultaneously. So what they do here, so basically they say, no, you know, actually, so, you know, you have the whole SNP variation, you have the phenotypes, you can't explain it, you should be able to explain it because we know that the trait is heritable. So what's going on? Well, you couldn't do it in this regression framework. You can do it in another framework, which is this. Right. What you do instead is you go back to quantitative genetics. Yeah, okay, maybe I'll explain it intuitively first. actually, let's go back here. The keyword, which are the 45% of variance, can be explained by considering all SNPs simultaneously. What was done before in all these calculations is that you find all the SNPs that are significant, one by testing them. So they're marginally significant, right? In just a regression sense. Then you put them basically into a sort of prediction thing in multiple regression, right? And see how much of the variance can all these things explain jointly as sort of a fixed effects in a regression framework. What these guys do instead is like, okay, let's not, remember those, the curves with parents and offspring, right? So we know that you can have a very high correlation if you just correlate relatedness on one axis, like, brothers are more similar in height than cousins that are more similar than second cousins and so on. And this fits a linear relationship, right? And the slope is the heritability, so relatedness between parents and offspring. That's exactly what they do here. So, but of course, there's no pedigree here. So this is done that was done by all those early Victorians in pedigrees, right? I showed you regressions between parents and offspring, but you can also show that the similarity in phenotype is dependent on the similarity in genotype, right? So basically, again, covariance in phenotype as a function of the relatedness, right? So with brothers sharing half the genome, brothers, sisters, cousins, and so on, all the way down, right? If you have pedigree data, you can do this in quantitative analysis. Cattle breeders have done that for a long time. You can estimate heritability this way. Okay, here there's no pedigree. These things are unrelated individuals, right? But of course they aren't unrelated. I mean, nobody is unrelated. I mean, this is obvious, right? Unless you're unless you believe in some kind of divine creation of everyone, we're all related. And in fact, otherwise, this is not one of many disproofs of sort of divine creation in that sense, at least, because you wouldn't be able to map genes if we didn't actually share alleles in common. We do, right? So that demonstrates that things came from that way. So what they do instead is that instead of trying to regress SNPs and phenotypes, they just say, let's use the SNPs to estimate relatedness between individuals. Right? These relatednesses go very far back, but if you take people in this room here, we are not all equally related, like I said, right? Some of us are more closely related than others, and in this case, it's going to reflect geography and so on, but even in a homogeneous sample, homogeneous, whatever, like all white people or whatever, there's still going to be these differences. And then you just look at the correlation, you regress the similarity in phenotype on the similarity in genotype as estimated by SNPs. So you use SNPs to estimate the kinship, the relatedness between individuals. And when you do that and basically regress it on that, you explain quite a lot of the variation. So more formally, what you do is, this is what I was going to show you, use a random effects model instead. You write the phenotype, Y, as U plus E, where both of these are random variables. This is the environmental noise. There's most models, if you do any kind of just standard linear regression, right? There is a normally distributed noise thing here with some kind of variance, and you assume that all the noise components are independent of each other, right? So you're all familiar with this. You've all done it. This is undergraduate statistics. This thing is slightly different, right? So here you say that this is also a random deviate. The phenotype of each of us is a random deviate for the mean, but, and there's going to be some kind of variance, and this is the genetic variance, but these random deviates are not independent. They have a covariance that is determined by the kinship matrix. So you basically put this into, you estimate the kinship between all the individuals, and you specify that there's a random deviation from the mean, and that random deviation, which direction you go, is determined by the relatedness between individuals. Then, then there is a sort of mixed linear model framework, or that you can use to estimate these variance components. There's a huge literature on this. It's fairly technical, and there's sort of math in understanding what you do. That's most just matrix algebra. Then there's some really clever tricks to figure out how to actually compute this efficiently, because people use these kind of models, but you can imagine that if you have sample sizes of 700,000, right? to actually estimate these things would involve like inverting a bunch of 700 by 700,000 matrices and things like that, which is not how you actually want to do things in a computer, but that's very technical. So, in other words, what I've been trying to say here, that the SNPs that are significantly associated with height in the marginal sense, right, one by one, jointly explain a tiny fraction of the variation. The joint effect of all SNPs, or even a reasonable subset, can be used to estimate the kinship or relatedness between individuals, and that explains a much higher proportion of the variance. So it actually so the stuff is in there. So okay, so what's going on? Well, basically, right. So this is a picture of Fisher. I mean, this is basically what Fisher would have predicted, right? So the reason this happens, now people have argued about this, but by far the simplest explanation of what's going on here is simply that the effects are really, really tiny. You have lots and tiny effects. You cannot significantly capture those. But as you see, I mean, you can try to estimate the effect sizes, and those papers I put up there, you can go and read how people are trying to do that, right? I mean, it fits, right? If you increase the sample size, you are going to find more and more, right? But even with 700,000 individuals, you still only capture part of it, right? There's a huge chunk that you probably will never be able to capture, even if you sequence everyone. And 700,000, that's bigger than the population of some small European countries, right? So it's you're getting pretty big. Right. Okay, so now with allele frequencies, yeah, so there's, so basically, on some level, to get this whole project going, there was a lot of hype going on, right, and wishful thinking. Either hype or wishful thinking, I don't know which one, but people, of course, wanted this to work. You're just going to crank this machine, and we're going to find all the allelic variants that causes, heart disease, then we're going to figure out what genes they are, then we're going to develop drugs and so on. So there were a lot of people who said that, this is absolutely ridiculous. So the idea was this, it was like, here's from some review article, I should, of course, this is one reason. I apologize to the author. You put slides online and stuff like that, right? I should, of course, have attributed this. I can't remember which article I stole this from. The idea here is that you have allele frequency on this axis. See what I drew here, and you have effect size here, right? Rare alleles, high effect. They cause Mendelian disease. You can map them in pedigrees and so on, right? Then the idea would be that you would be somewhere here. You'd have common alleles with smaller effect, but they would still have a pretty significant effect because they would sort of be so common, right? And then, of course, there'd be very few examples, but I'll show some examples where that's not true, where you have common variants that also have a big effect, right? But the idea would that you be here, and here you do Mendelian thing, here you do GWAS, right? Here you're in trouble, right? If you have rare variants of small effects. Okay, so here is an attempt to estimate this. This is more like what the curve actually seems to look like. This is also a couple of years old, but an attempt to basically look at the effect sizes as into allele frequency. I think it was Andy Clark who once called this whole thing the world's most expensive test of the mutation selection balance hypothesis, right? So basically, you can ask, and people did ask, why would there be like disease alleles that are common in the population? Well, the idea said, well, most of these diseases that we have today are only an effect of us having lived so long, we eat too much, blah, 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 so they were not selected historically. Based on what we see now, that probably wasn't the case. They are deleterious, they were deleterious, and they are basically following a curve like this for most diseases. Yeah, so then there were Cassandras. You're familiar with the story of Cassandra? This is from the Iliad, right? Cassandra was a priestess she'd been cursed by rejecting the advances of Apollo, the god of prophecies and so on, right? So he cursed her by basically being able to foretell the future, but nobody would ever believe her, right? So she can be sort of a feminist icon. There's actually been books written about this. Nobody ever listened to her, right? She always knew what was going on. So she predicted all the disasters and so on. Anyway, so there were people who basically said, remember the paper I showed, how many SNPs does it take to map a disease, right? So these guys turn it around. How many diseases does it take to map a gene with SNPs, right? How many diseases will actually have the property that you can map things very well? Now, of course, they were also slightly wrong because for all this stuff that's been done, there's no question that we're learning a fantastic amount of diseases, right? So what was clearly wrong with this entire endeavor, we are not at the case where you should just take SNP and predict whether someone is going to get sick or not, right? With very few examples. I would not be scared by putting my genome out online. Who cares? You don't learn anything from that, right? It doesn't work like that. What is true is that these hits you get in particular genes actually do tell us something about the biology of the disease, and that will, I believe, over time lead to diagnosis and so on. So in that sense, it does work, right? What could work? In some sense, prediction. Prediction can work in a statistical sense, though. An insurance company, the prediction value, given how much of the variation you explain, is so little that using it for individual diagnosis, like personalized medicine in that sense, right, does not work, in my opinion. So if you have any stocks like that, sell them. But of course, an insurance company could use it very well, and a cattle breeder can use it, right? Because if you can only have 3% prediction, well, that can be your entire profit margin, right? So if that's what you're doing, that's a different story. But you would never kind of recommend surgery not based on 3% margins. Okay, right. Okay, so I'm going to talk. So that was human stuff. Now I'm going to move into talking about population structure, and I'm going to talk about non-human stuff. So any questions before I go on here? Yes. Well, no. Yeah. Yeah. No, so in most of these models, they assume they're independent. I'm gonna, there's also questions about epistasis and stuff like that, right? So, the kinship is about, the kinship is about not the correlation. Yes, correlation due to ancestry. So basically that's one way of looking at it, of course, from a quantitative point of view, that basically because, yes, exactly. You are more similar to your sibling because, of course, you share a lot of things, so they are in fact correlated. So the joint effect of all these SNPs you can estimate, right? That's correct. And by using but you're not looking at the causative SNPs most of the time. You're using SNPs across the genome that have nothing to do with the phenotype, just to tell you what effectively, right, you estimate how much of the genome do you share in common using sort of random markers. And that also is a proxy for how many of the causative SNPs you share in common, right? And that basically, because you share more in common, you will therefore have a more similar phenotype to somebody you share more SNPs with, right? But I was sort of talking at your poster about this, whether this works or not. That, of course, for that to work, that is absolutely true in the infinitesimal model, because the effect of each SNP is going to be very weak, so that they will not. But of course, under selection, like stabilizing selection or any other selection, neutral SNPs across the genome will not necessarily predict what the causal SNPs are doing, right? And there's some interesting research. That's a research area, right, to figure out what that happens. Then I think you can actually learn something about the architecture of traits that way. And Nick and I talked a lot about this, so this is something we can talk about separately. Okay. Right, I'm going to talk a bit about population structure here. Okay, so this is while all this stuff was going on in human genetics, people doing GWAS in model organisms, and there weren't very many because the resources weren't there, had run into a very different problem. And I'm going to have to speed up a little bit here. So first, this is one of the early GWAS outside humans that was done from Arabidopsis that we did. And you see here, these are now 5 chromosomes, otherwise looking the same. And this is a sample size of 96 individuals. And you have these beautiful spikes here. And you see, these turn out to be all particular pathogen resistance. This is this gene for gene thing that was being discussed by Santiago. Like basically we're measuring the response to a particular bacterial pathogen and bang, you just find the gene, right? So this is an example where, you have really, really big effects. So whoa, it works. Okay, great. And furthermore, and if you zoom in here, this is kind of what I'm saying, you see one of these peaks. Now we zoom into a scale of, what is this? This is KB scale, tens of KBs, right? So you basically, and the blue line here is right where the gene is. So beautiful, great, okay, fine. But it doesn't always work like this. This is the one I just showed you. We also found cases where we didn't find anything because, of course, with such small sample sizes that we could afford to do, the trait has to be very, very simple to find something. But that's not one. That's obvious. That's nothing different from what I said before. Yeah, and this is like a significant threshold here, right? Okay, but what we quickly discovered, and this was also the people who did mice and maize discovered, is a very different problem, and which you can see here. If you do a genome-wide association for flowering time, so basically you take plants from all over the world, they are adapted to read the climate in a different way. So you put them together, they will flower at very different times, and then you do a GWAS on that. And you see here's that significant threshold, and bang, there are peaks everywhere. And if you were to sum these things up, so these are all nominally significant, they explain like 4,000% of the variation, right? Obviously not, right? They're obviously not independent. What's going on here? It's actually pretty obvious. This has to do, let me see, what's my next slide here? It's basically because of population structure. Another way to see it is like if you look at the p-value distribution across the entire genome here, right? It should be flat. Like, 5% tests should be significant at the 5% level, but you see here, in fact, 30% of the tests are significant at the 5% level. So what's going on? Well, basically, this is alleles are not independent. Here you're looking at something where selection has put alleles in very different populations. So we run into the same problem. So I was also involved with an early study to look at dogs. So we had dog breeds of all kinds, right? And they had found that insulin growth factor 1 is a very important determinant of dog size, right? So you get these kind of things. And then you ask, can you estimate the effect size of of the IGF-1? How much of the variation does it explain? And of course, you can't do that, not from a population sample, because selection has this guy. So IGF-1. These different alleles does explain some of the variation, but these guys have big alleles at lots and loci across the whole genome, and these guys have little alleles across the whole genome, right? And they are in extremely strong linkage equilibrium because of selection across the whole genome. So if you try to estimate the effect of one of these, you can't, right? Because they're completely confounding each other. Each one is doing the effects of everything else. And furthermore, there will also be a bunch of other SNPs that are just correlated because of just of history, right? So if you took, just look around the room here. Like if you did the thing I said about trying to map eye color, right? If you did that, if you're like a human geneticist from outer space and you landed on Earth and just rounded up a bunch of humans, and sequence them and look at who had blue eyes and who didn't, and you found some alleles for that, you'd find them, of course, but what else would you pick up? Blue eyes is correlated with hair color, it's correlated with skin color, it's correlated with body mass index, it's correlated with malaria resistance, it's correlated with SNPs across the entire genome just because of human demography and migrations, right? I mean, there's population subdivision. So obviously, if you then do a test by this, a lot of things will show up as significant, even though they have absolutely nothing to do with the trait, right? Some of them will be other selected genes that are in association. Some of them will just be there by accident, and you have to figure out some way to get around this. Okay, so then basically what we, and this is mostly the person who deserves most credit for this is Ed Buckler, a maize geneticist, figured out that you can handle this using the same mixed model as was done before. Phenotype again, sorry, I switched notation here slightly, but you can live with that. I think at least I'm using. So here's phenotype, right? And then you're testing a SNP effect. Like this is standard, and this is really a regression coefficient, right? This is a matrix notation here. But the phenotype, you also put in exactly what I had before, a random deviate. That is due to this general kinship thing, and then environmental noise. And if you fit this kind of model, so what is done in these, when people were trying to, for the human height data first, is exactly this, but, excluding this thing, right? So just linear regression, right? Regression coefficients, one SNP at a time, testing it, and environmental noise, and then you can get association. Here we put in this variable again. So why do we do this? Because basically, in order to see whether any particular thing is affecting it, we first say, let's see how much overall genome-wide relatedness explains. And sort of let that part of the variance be soaked up by this sort of background effect, right? Which we're not trying to estimate, and then see, are there any things that are significant on top of that, right? And that works. But it's, of course, in some sense, extremely conservative, because if you did... So for the eye color, I just sample, I think it would work on a human, if you had a sort of a global human sample. But say that you did this exercise, and you had a sample of only like 100 white Swedes and 100 black Nigerians, and you put them together, and you put this framework, kinship would explain almost everything, right? And you know, because basically the trait is like fixed between the two populations effectively, right? And you would like throw out the baby with the bathwater. You would not be able to map anything. Okay, so let me see what's the next thing here. Yeah, so basically, so that kind of model works if you, and it is being used routinely, and it's actually, yeah, so here I put this up that you can read for yourself, right? There were other attempts. People sometimes say that to deal with population structure, what you need to do is like actually model it. You take the population structure in as a cofactor and stuff like that. But actually, you know, the very real problem, I mean, you can get false positives due to population structure, but that way you will never miss the true ones, right? The very real problem is the one Kavita alluded to, that you have linkage equilibrium between all the causal sites. What you really would like to do is, I mean, the reason we run into problems though this model is that we're doing marginal testing one thing at a time. If you could simultaneously fit all the causal alleles, right? You know, then that wouldn't be a problem. Then you know, the fact that they're correlated, you can take that into account. You would know that you can't, that they're not dependent from each other. But of course, this is in practice. In theory, that's what you like to do. In practice, this is not an easy thing, and we have like a review thing about this, right? Okay, let's see here. How am I doing? Do I need to... So I have it till 11, yeah? Yes, so 20 more minutes. Okay, there is one more thing I wanted to say about, so population structure leads to these genome-wide problems of finding things, but there is another problem which is tricky and which is a bit interesting. It can lead to real problems with fine mapping, and that's important if you ever do this. So for instance, I don't know if anyone, I mean, so for instance, it shows up in GWAS for rice all the time. And I don't know if anyone here is interested in rice, but there is actually, I do know that. So I think I should go through it. So genome-wide confounding, a problem, and we know how to deal with it, sort of. This one, there's not a good solution to yet. So here is a GWAS for germination rate in Swedish Arabidopsis. So, and you find this nice spike here, and it looks like it's right over a gene that is involved, that is known to be involved in regulating seed dormancy. Okay, so great, wow, yay, we found something here, right? But now when you look carefully, you see something like this, and this is something that's been seen in almost all GWASs in Arabidopsis, and in rice, and in lots of other organisms, and you should know about it. Basically, if you now zoom in on this peak, it doesn't look, remember that bacterial resistance thing I showed you? That was a spike, that was a SNP right in the gene, beautiful, yay. It doesn't look like that here. Here, if you zoom in on that nice spike over this scale here, which goes from 18,560,000 to 18,610,000, right? So this is on, you know, this is like tens of kb's here, right? You see that the actual tip of that peak is sitting over here. And actually, it's not like a nice needle-like thing like we looked before. It's more like a mountain range, whoops, with, it's more like a mountain range here with like lots of different peaks. And the obvious candidate is actually sitting over here. Here is this, the spike. You know, next to it is a gene that is involved in embryo development. So, you know, if you didn't know what you were doing here, you would say, well, this is surely the most likely candidate, right? Because it's right next to the spike. Turns out that it almost certainly, and we have shown this now, this one is the one that's actually doing it. And you can see this by, excuse me. That's something you should stretch from there. if you put in 2 SNPs in the promoter region of this thing as cofactors, this entire mountain range here effectively collapses. So what's going on here? It's again a case of using the wrong model. There are multiple alleles of this gene in the population. There are 2 different SNPs. They're sitting on different haplotypes. Both are involved in affecting dormancy. I think they're leading to, did they cause dormancy or loss of dormancy? I don't remember now. Doesn't matter. Okay, so there are 2 different things. What the standard GWAS approach does is take 1 SNP at a time and ask, is it strongly correlated with dormancy? Right? But there isn't, but that's the wrong model, because there are 2 different causal factors here, right? And you're going to try to fit one. Is the model's misspecification, right? What's going to happen? Well, you're going to pick the SNP that is the best. The one that's going to get the strongest association is the one that best captures the joint effect of these 2, right? So it has to be in linkage equilibrium with both of the 2 other ones, right? And effectively, this is a phenomenon very similar to, I don't know if, have you ever heard of ghost QTLs? So when people did QTL mapping, mapping different modes, they often found that if there were 2 closely linked genes in a linkage mapping experiment, the highest peak would be somewhere in between them. Because basically, the likelihood, since they're fitting 1 gene at a time, you're going to get the strongest association between this, because that captures sort of the effect of both of them, right? And that problem was solved by basically doing a regression thing, that you find 1 peak, then you put that in as a cofactor, and then you try to find a new one, and then you can remove the ghost and see that there are actually 2 things, right? And this is actually the same thing going on here, but with linkage equilibrium, it's much more complicated, especially if there's been selection, so there's like long haplotypes and things like that going on here. And you will get a spike somewhere else. So here's something else that would be actually, there's room for some work to figure out how to take apart these things in a good way. We don't know how to do this yet. We've seen it everywhere. And if you think about it, if you have allelic heterogeneity as some selectively important gene, right? And that turned out to be very, very common. Then when you fit individual SNPs, you're most likely to pick something in the vicinity that is highly linked, but it's sort of just the proxy to both of them. In this case, because we had a good candidate, you can basically start doing things. Let's test putting in other things several at a time and see if you can disentangle it. Yeah, okay, so here's a simulation example, just to bring this home very vividly. We simulated allelic heterogeneity at different loci, because this doesn't only work between, right? There's so much linkage equilibrium in some of these populations that you can get things wrong and even on different chromosomes. So here we took Arabidopsis data, and we said, okay, what if there's a trait, like a disease, and we pick SNPs sort of at random. Either one is sufficient to give you the disease, so they're strongly epistatic, right? They're not additive. Either one will give you disease, right? So then you have the disease for 2 different causes, and you just try to find the true one. And we quite often found cases like this. So here in the simulation here's one causal locus, here one causal locus. When you do a GWAS, in this case, you find one of them, you miss the other one, and you can find a false positive on the different chromosomes. So this is a very nasty thing that can happen. And one reason we often complement our GWASs with crosses and things like that to make sure that you're at least in the right vicinity. Okay. Yeah, okay. So things you can do. So here's a paper we wrote a couple of years ago. You can do GWAS with multiple loci at the same time. So what's badly needed is models that don't fit one thing at a time, right? And there's a lot of work. It's very, very hard on try to do a fully Bayesian estimation where you fit all effects simultaneously, because that's really what you would like to do. So there's theory on this. Okay, yes. I was going to end on, yeah. Okay, so I showed first that, so we found that GWAS works really well in Arabidopsis, and there was a write-up that basically said plants are not like humans because you find things. But I just want to point out that this should have been the title. The reason we find very different results in our Arabidopsis stuff, and the same thing in maize and so on, is not that there's anything very unique about plants versus human. It's simply it's the genetic architecture of the trait, and that, of course, is a consequence of selection. We are looking at, you remember that curve with, you know, a slope like this? I can just. So if you have frequency and effect, and like for many of these traits like height and so on, it kind of looks like this, right? Things that are common tend to have a small effect. Things that have a big effect tend to be very rare, and diseases in general are even more like this. You know, height will look like this presumably because it's mostly under stabilizing selection, and very rarely do you find things out here. Well, we do find things out here. Where would you find that? Of course, because selection maintains variance, right? Then, you know, for instance, because of local adaptation or things like this. So one thing I always went around saying that the flowering time stuff and so on we see in Arabidopsis, all the confounding, the big effects, that's nothing to do with plants, it's all to do with traits. If you looked at a trait like human skin color, you would see something very different. And so I got an opportunity, and this turns out to be true, I got a chance to be involved with one of these skin color experiments, and there's been more on this more recently. So this was a paper done in the Cabo Verde Islands. So this is off the west coast of Africa. Basically, it was a center for the African slave trade, and basically complete mixing of West Africans and Portuguese and probably a whole bunch of other people over hundreds and hundreds of years. And as somebody put it, they're all poor, so there is no sort of caste, skin color kind of nonsense going on. Everyone is just poor fishermen, and they have just been doing what humans do for several hundred years. And you get all kinds of cool-looking people, right? So yeah, so here you can see, you know, you can obviously disentangle eye color from all the other phenotypes. You get very beautiful phenotypes. And so people have gone out and measured both skin color and eye color and so on. If you measure skin color, you actually, you know, you do it underneath the arms, right? So it's mostly not so heavily affected by the environment, because otherwise, of course, your skin color depends on how much you're out in the sun and so on. And bingo, it looks a lot like this. So this is a small sample size of only 600 individuals, and look, massive p-values here. Yeah, these are just different. This is with and without various forms of correcting. So this is correcting for, this is without correcting for population structure. This is correcting for population structure. You know, significance goes down a lot, but basically you still have the same peaks. And these are in gene. This is tyrosinase. These are genes that one of them is the agouti locus in mice, right? They're involved in mammalian skin color proclamation. Clear candidates, right? Oh, I forgot to say the other thing. There was a paper by Jonathan Pritchard recently who sort of pointed out that as we increase the sample size in human GWAS, we don't actually learn so much about the traits, because it seems like a lot of the genes involved are not terribly involved with anything, right? And that's probably because once you start fishing into these really small effects, it's just pleiotropic effects across the entire genome, right? The big effects, I mean, here is very different. I mean, these genes are definitely involved directly in the trait. So you find them, okay, so, and now, guess here, so this is a small sample size, there were only 600 individuals, we found 4 loci. How much of the variation do you think you can explain, or skin color? With just 4 genes. Just a guess. This is a mixture between Africans and Portuguese, right? So, no one dares to guess. There's no really true or right answer here, right? It's clearly going to be more than for human height, right? It turns out to be about, I think it was like 32% or something like that, which is, you know, quite a lot with just these genes. But that's interesting because, in some sense, this is like the model. I mean, so here, so even for, yeah. So you can, to some extent, predict skin color with just knowing these allelic variants, right? And there's been a couple of papers recently that try to reconstruct, you know, you could do this with genome sequence, right? So I said that you can't predict disease and stuff like that, but you could actually, if you looked at my genome, you could predict that I had blue eyes, and you could predict that I had fair skin. Those things we actually know how to do. But actually, only partly by looking at these genes, because it turns out, let's see if I have this here. Yeah, this is cute. So there was like 32%, if I remember it correctly, maybe it's 35, that you explain with these 4 SNPs. But actually, if you pick just 500 random SNPs across the genome, you predict phenotype even better. And that's because of this population structure confounding effect I said before, right? Because effectively, then you just predict the fraction of African versus European ancestry. That you can do very effectively. So this is the whole population structure effect, and this is what Kavita was saying. You know, of course, the reason they have different skin color, in this case, is of course that there are genes involved, right? Most of them are too small effect that we can find them. There's a couple of big ones that are clearly segregated, and we can find them whether we correlate correct with population structure or not. And there's a long tail of smaller effects that are clearly segregated in allele frequency between Portugal and Africa. You put them together, you know, they're still in linkage equilibrium, and you can sort of identify these blocks, and you know, that's why you can tend skin color. And good, I think that's all I had to say. So any questions about this? That was my last slide. Questions? So, well, I can say, I mean, so these kind of, in these early case control settings, population structure for humans, that wasn't such a big deal, right? Because you basically take a population, you have a clearly defined phenotype, you take a sort of ethnically, demographically matched sample. Human epidemiologists are strongly trained to figure such things out, right? And you just compare the allele frequencies. But now, increasing what happens, if you've seen like the UK Biobank and stuff like that, where all this thing is going to is that people genotype the entire population and cross-references with a whole bunch of things. And then, of course, actually, you know, demography and all this stuff does matter. Like there's been several studies on the Finnish population, and they're clear, you know, allele frequency gradients going from Eastern Finland to Western Finland, depending on the admixture between Swedes and Finns, for instance. So methods for taking all this into account simultaneously, right? So you have demography, you have multiple causal sites, and trying to puzzle this out are really very important. And there's like an enormous amount of work being done, and a shocking amount of data. You know, this UK Biobank stuff is public, right? You can just look into it if you want, right? And all our data are publicly available as well. There's also, you were mentioning Trudy McKay's website for the Drosophila thing. So for Arabidopsis, we have sort of a similar thing. So all these lines, there are 1,000 lines for Arabidopsis, you can just order them, and you can just upload the phenotypes, and you can basically run interactively GWAS on the spot and see what happens, and even play around with conditioning on different sites and so on. Questions? Yes, Fiori, any questions? All questions? Mm-hmm. Mm-hmm. You tell me. I think you know better than me, actually. Yeah. 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 Yes. Yes. So so one of the things that, I mean, so Peter Vischer, that this of course is trained in Edinburgh and all that, people have been trying to work it, but there's much more work to be done. I really think that to try to figure out what is the, I mean, there are all these models, House of Cards models, you know, all this stuff, right? It needs to be revisited to look very carefully at what's going on here. And I, you know, personally, so a question I'm really interested in is this question I was bringing up, but your poster up there, right? Basically, you know, looking at in the human stuff, right? I mean, they assume this infinitesimal model. You estimate the kinship based on genome-wide markers. But what you're actually, that's just like a proxy, right? Because what you actually would really like to do is fit all the causal sites at the same time, right? Forget about the non-causal stuff. And they may have a different distribution. And they say, oh, I didn't say that actually, since I have a little bit of time. Another cool thing that's been done is, this is done in both humans and in mice. If you can take subsets, you can have two different random effects, right? You can estimate, you can say, for instance, okay, let's ask the following question. Let's take all the genes in the genome and all the promoter regions and look at all the polymorphism there, estimate how much of the variations accounted for these things compared to the rest of the genome, right? And then you can see, I think, both in mice and in humans, that the coding regions plus open DNA regions explain like 70% of variation for a lot of quantitative traits, and the rest much less, right? So you could have partition variance between different parts of the genome as well. That's also very interesting, right? Because, right, I mean, that's the whole genome contribute equally into. There are lots of really cool things one can do here. But yes, from this, right, one should people who works on these quantitative models of selection should really try to dig into these data and see what we can learn about what the architecture is, I think. Okay, coffee, I guess, yeah.